Hello, my name is Sean McIlwain. I work as a scientist at the University of Wisconsin-Madison School of Medicine and Public Health. My talk is entitled MASH Native, a universal and comprehensive software for native mass spectrometry and top-down proteomics. Top-down mass spectrometry is a powerful method for comprehensively characterizing proteoforms, which arise from genetic variations and post-translational modifications. However, a paucity of data analysis software designed for top-down MS is recognized as one of the major barriers to utilizing top-down proteomics. Native top-down MS is even more underserved than denaturing top-down due to challenges in deconvolution of low-resolution MS data and poorer fragmentation efficiency than denaturing top-down MS. While a number of software tools have been developed to address certain aspects of native top-down mass spectrometry, individual tools are limited in scope and MS file type compatibility. To address this challenge, we present MASH Native as a universal and comprehensive software environment for native top-down proteomics. Written to run within the Microsoft Windows environment, MASH Native is a descendant of MASH Explorer and provides an intuitive graphical user interface. After data import, the user has access to chromatogram and mass spectrum views. MASH Native guides the user through top-down data analysis and validation. MASH Native includes support for multiple vendor formats, integration with many algorithms for deconvolution and database searching, and also performs the necessary conversions between upstream and downstream tools. Recently, UNIDEC has been incorporated into the suite of deconvolution methods, which significantly improves our native top-down handling abilities. MASH Native provides both automatic and interactive modes to allow users flexibility in running UNIDEC within MASH Native. MASH Native provides the ability to search both terminal and internal fragments. To help with visualizing the internal fragment matches on the sequence table, we have also provided heat map coloring of the amino acids to indicate the amount of fragment coverage. The utility of internal fragment searching for native top-down MS analysis is clearly seen in a recent publication from the Go Lab, which directly infused native cysteine-linked antibody drug conjugates, or ADCs, in a timsTOF Pro mass spectrometry for a three-tiered MS analysis. First, the collisional cross-sectionation of the intact ADC was measured by TIMS. Then the intact mass was determined by time of flight MS. The detected ADC could be deconvoluted using UNIDEC to determine glycoform state and average drug to antibody ratio. Non-covalently associated subunits were released from the ADC by in-source CID, then fragmented using collision cell CID. Because the intratane disulfide bonds are intact during MS analysis, the release of the subunits is not complete. The fragmentation efficiency of subunits and resulting sequence coverage is low when only assigning terminal fragment ions. When searching internal fragments, the sequence coverage dramatically improves, and fragment ions generated from within disulfide bounded regions are revealed in the light change with one bound drug, which is LC1. This enabled broader sequence coverage and localization of disulfide bonds within LC1. MASH Native also allows users to perform spectral summing in a variety of different ways, including several options for selecting which scans to average, which algorithm to use for performing the summing, and how to handle MS1 scans. MASH Native provides additional tools, including an ion finder that can help with the discovery of putative ions from a proposed proteoform that may have been missed by the deconvolution algorithms. MASH Native is available through our website and has been downloaded by users all over the world. We have many avenues for users to communicate with the MASH team, including a forum, Twitter, and email lists. In summary, MASH Native is a universal software platform for analyzing native and top-down proteomics data. I'd like to thank our collaborators, the past and present members of the MASH team, the Dr. Inga lab members, and our funding sources. MASH Native is actively maintained and continually improved upon to be an indispensable tool, and we appreciate your feedback. Thank you for your time, and I hope you consider giving MASH Native a try.